Hello to our friends joining us via recording. Today we are starting unit number 3. Unit number 3 only has 3 lessons in it, but don't let that fool you. There is just as much content in unit number 3 as there have been in the other ones that had 4 lessons. So make sure to give yourself plenty of time to work through lesson number 9 and lesson number 10 especially. We're going to cover a lot of lesson number 9 today. We'll wrap it up next week and hopefully start lesson number 10 next week as well. So lesson number 9 is all about the muscular system. Muscular system being what we use to move it. So to help us remember from what we talked about in lab about how we move our body and our muscle tissue, we did some group work together. So the first thing we did with our groups is we looked at our 3 types of muscle tissue, which, by the way, will show up again on the lab final exam. They will also show up on the unit number 3 exam. So that's good news for you. We already know how to identify these tissues. That'll help. Before we start matching up some of these descriptions of lab-based stuff here, let's go through and work on identifying our types of muscle tissue. So we'll work from the top down. This first type of muscle tissue that I see here in the top image, which kind of muscle tissue is this up here? Yeah, so this first type of muscle tissue that I see at the top, I guess I'll type it on there, is cardiac muscle tissue. Cardiac muscle tissue. There are a couple of different features of this tissue that help us to recognize it. Let's start with these, because I didn't actually include them on our list here. These dark lines that you see right here, do you remember from lab? What did we call those special dark lines? Yeah, look at you all. You're fast typers. Those are those things called intercalated discs. Intercalated discs. Remember, this is the way that cardiac muscle cells attach to each other. Here's a let's tie it together moment. Intercalated discs, these dark lines that we see right here, these are actually a whole bunch of desmosomes. Desmosomes. Let me type that word out again. Does anyone remember when we talked about desmosomes? Where have you heard that term before? Anybody remember? It's been a while. Yeah, we talked about them being anchoring, right? Yeah, they're my type of attachments. We talked about them in the skin with those keratin proteins. So we might call them anchoring junctions. They have all those proteins that make them strong. So this was the one I kind of mentioned to you, like if you go into, exactly like Jalapeno said, if you go into Walmart and you look up at the ceiling, you can see all those metal bars that are kind of supporting the ceiling. That's what those desmosomes looked like. They had those proteins that held them in place, made them a really strong attachment site. So cardiac muscle tissue, these dark lines that we're seeing here, are actually desmosomes, helping us to hold these types of cells together. That's a good thing that we have really tight anchors to hold stuff together, because remember that cardiac muscle tissue is always contracting. This is what's going to give us our heartbeat. This is what we find in the heart. So we want to make sure those cells that make up the heart that are constantly contracting, we want to make sure that those are anchored together really well. My first type of muscle tissue that we've identified, cardiac muscle tissue. What's this second type of muscle tissue that we see down here? Who's type number 2? Yep, type number 2 there, that is my smooth muscle tissue. Smooth muscle tissue gets its name because it is striated or non-striated. Which of these words applies to smooth muscle? Striated or non-striated? Yeah, exactly. Here, we'll draw our first line here. Smooth muscle tissue is my one type of muscle tissue that is non-striated, meaning it doesn't have those stripes. It's a little hard for us to see in this particular picture of cardiac muscle, but if you squint a little bit, you can see some stripes kind of over in this part of the cardiac muscle tissue. It's very obvious when we look at our skeletal muscle tissue down here, that we've got those stripes, those striations. Smooth muscle tissue is the one type of muscle tissue that does not have striations. It is non-striated muscle tissue. When we talk about smooth muscle tissue, our cells also have a special kind of shape. Did we figure out which kind of shape of the stuff that's listed up here, which shape word applies to them? Yeah, it's that weird spindle word. So we would say that smooth muscle tissue is spindle shaped. I know I saw at least one of my groups drew what that looked like. So you can hear it called different things, spindle shaped or fusiform. When we look at these muscles, they've got a wide part in the middle with a nucleus and then two really skinny sides, the individual cells. So we come over here and look at, we can see those nuclei really big, and then this cell kind of stretches around it. And here's another nucleus, and we're kind of stretching around it. You're not going to really see the spindles super clearly in smooth muscle tissue, but you're definitely going to see those nuclei. We can see those enlarged nuclei all over the place in the tissue. Spindle-shaped cells here in smooth muscle tissue. Our last type of tissue we mentioned in passing is our skeletal muscle tissue. Skeletal muscle tissue. Remind me, we said that skeletal muscle tissue is or is not striated. Is or is not striated. Yeah, it is. Several of you beat me to it and you're like, I can type the whole striated word. Look at me. So skeletal muscle tissue is striated. It's the type that's easiest to see. While we're at it, talking about striations, those stripes, we'll go ahead and add that up here to my cardiac muscle. Cardiac muscle is also striated, also has those stripes. Today, we'll actually talk about how we make those striations. It has to do with the way that we arrange our proteins that do contraction. So the order that we put them in leads to these stripes that we can see. When we talk about skeletal muscle tissue, is this one long and cylindrical or branched? Skeletal muscle tissue, long and cylindrical or branched? Yeah, these are the long ones, right? So I'll draw my little line down here. And you can see that when we're looking at this image. Notice how this cell looks kind of like a tube. It's a long, skinny tube. It's got those stripes on it. That's what we see in skeletal muscle tissue, long and cylindrical cells. Compared to cardiac muscle, which we would say is branched. Remember when I think when I drew a sketch for you of what cardiac muscle tissue looks like, it sometimes kind of looks like the letter Y. So you've got kind of Y-shaped cells that branch and then they connect to each other. So we end up with this branching, and at the branches you'd find those desmosomes. So Y-shaped or branched cells, that's my cardiac muscle tissue. Long and skinny cells, that's my skeletal muscle tissue. And then we can't even clearly see the cells really with smooth muscle, but if we could see them, they'd look a lot like this. So here's some basic anatomy of our muscle cells. Now let's briefly mention the physiology of our muscle cells. When we talk about the type of muscle tissue that moves bones, which of my types of muscle tissue is really good at moving bones? What helps us with movement, essentially? Yeah, movement is skeletal muscle. So friendly reminder that this is my skeletal muscle down here. When we're talking, yeah, so Elapina mentioned something, we'll add it now too. When we talk about moving the bones in your body, so making your body move, that would be what we consider a voluntary action. You decide to take notes. You decide to type. You decide to look out the window, right? Because you're bored. Hopefully not too bored. But you decide what you're doing as you move the bones of your body. That's considered voluntary muscle contraction. When we talk about the movement of blood in your body, when we talk about the movement of food in your body, those are things that we're grateful that we don't have to control. When we talk about moving blood in the body, What kind of muscle tissue helps to move blood? Oh, we got a couple answers here. Oh, Kira, you backtracked on yourself. You're not actually wrong. So the obvious answer, oops, let me get my writer back out here. The obvious answer would be cardiac muscle tissue, right? Cardiac muscle tissue, where do we find that again? Where's cardiac muscle tissue found? Yeah, that's found in the heart, okay? So the heart is really important in moving blood. But yeah, what Kira mentioned before, smooth muscle is also involved in moving blood. One of the places that you find smooth muscle is inside blood vessels. Blood vessels. So when we talk about your blood pressure, what we're actually talking about is how contracted your smooth muscle inside the blood vessels is. So when we talk about moving blood, there's actually two types of muscle tissue that do that. When we're talking about specifically moving blood kind of in the center of the body, around the chest, for example, that's the job of the heart. It's cardiac muscle. But everywhere else in your body, we use smooth muscle in the blood vessels to make sure that the blood keeps moving. So there were actually two answers for this one: cardiac muscle and smooth muscle. Yeah, like Fanshin mentioned, cardiac muscle is going to pump it, and then smooth muscle is going to keep the pressure to a point where the blood keeps moving. Absolutely. When we talk about moving food, which kind of muscle tissue helps us to move food? Who's in charge of that? Everyone's favorite, right around lunchtime, right? Exactly. Yep, that's going to be the job of my smooth muscle tissue. Smooth muscle tissue. This smooth muscle tissue that I find in my favorite system. Who remembers my favorite system? We haven't talked about it much. Yep, find the digestive system. That's my favorite. So, smooth muscle in the digestive system helps us to move food. Smooth muscle in the blood vessels helps us to move blood. Remember though, we kind of hinted at this, when it comes to our types of muscle tissue that move blood and that move food, both of those, cardiac and smooth, both of these types of muscle tissue are involuntary, meaning you do not decide when they contract. And again, when we first talked about this in class, we talked about how that's a really good thing, because that would be your entire existence is making sure your heart never forgot to beat and making sure your blood vessels and your digestive system always contracted when they needed to. So we're really glad that we don't control these ones. This skeletal muscle is the one that we do control, and that's the one that allows us to do movements. I'm glad that you all found this question. I forgot to intro it for you before we split into group work. We always talk about how Dr. Ollis does tricky questions on the exams, right? So I wanted to give us some practice with a tricky question, and we're going to kind of dissect it together to way through it. This question is talking about someone having an asthma attack. Now, I chatted briefly with one of my groups. When a person has an asthma attack, they are or are not in control of the muscles that are giving them trouble. Do you decide to give yourself an asthma attack? Yeah, probably not, right? Kira's joking. Oh yeah. I mean, if you get stressed out enough, you could probably trigger one. But when we talk about an asthma attack, what's happening are the little airways in the lungs called the bronchial tubes. They're getting smaller. The amount of space that there is for air to get into the lungs is actually shrinking. This is because one of our types of muscle tissue is contracting, making the size of our normal bronchial tube, here's normal size, our normal size of bronchial tube gets a whole lot smaller. The other thing that happens with it too, you'll probably talk about this actually in A&P2, the other thing that'll happen then too is as the tube gets smaller, you still have mucus that lines these tubes, except there's not as much space, so the mucus feels like it's thicker. So someone having an asthma attack, we went from having a lot of space for air to go into to very little space for air to go into. When you're having an asthma attack, albuterol is a medication that you use a lot of times to undo this, basically, to increase the size of the bronchial tubes again. So when we talk about the way that a rescue inhaler works, what we needed to figure out as a group is which kind of muscle tissue it's working on and what it's doing to that tissue. So let's start with the second half of our sentence here, because that's where I think a lot of my groups started here. If we're going from my little tiny airway to this much larger airway, which is what albuterol does, we go from little tiny to much bigger, would that mean my muscles contracted or relaxed, going from here to here? Yeah, that means my muscles are relaxing. So the job of albuterol is to get some muscles to relax. If they relax, the space opens up. There's more room for air to pass through. Now we need to figure out which kind of muscle tissue this acts on. Now, one of my groups was toying around with the different types. We immediately got rid of cardiac as an option. Why can we immediately knock out cardiac muscle tissue as an option? How do we know this one's wrong? Yeah, Fanshan's right. Cardiac muscle tissue, literally the only place we find that is in the heart. So we know that this cannot be cardiac muscle tissue. We're talking about the airways, not the heart. So now we have to decide if we're talking about skeletal muscle tissue or smooth muscle tissue. And this is where we come back to what I mentioned at the very beginning of us looking at this question. When we're talking about an asthma attack, a person doesn't decide to make their airways small. Their airways get small all on their own because they smelled the pollen in the air or because they were exposed to something that irritated them. When someone has an asthma attack, this is involuntary muscle contraction. Involuntary muscle contraction. If it was voluntary, they'd be able to breathe all on their own, right? They could just decide to stop having an asthma attack. But in an asthma attack, they're not in control of the muscle contraction. So we're using albuterol to relax those muscles. When we talk about skeletal muscle and when we talk about smooth muscle, which one do we not control? Skeletal muscle or smooth muscle? Who are we not in control of? Yeah, exactly. We are not in control of our smooth muscle. So way down in your bronchial tubes, if you remember from lab, we said that smooth muscle, you find it inside hollow organs. The bronchial tubes are hollow airways for air to come inside. Down inside of them, we have smooth muscle tissue. When smooth muscle tissue contracts on its own at the wrong time and too much, we might need some albuterol to relax it, to allow us to be able to breathe. Okay, this is an exam type question, right? So I would give you this kind of information and ask you to fill in the blanks, just like we did here. Here's the stuff that you use to answer this question. First thing that we needed to know when we were answering this question, we needed to know where we were at. So we're talking about the bronchial tubes. Even if you don't know for sure where the bronchial tubes are, kind of like we talked about, we could eliminate cardiac muscle as an option because cardiac's only in the heart. So I need to know my locations of my muscle tissue. Cardiac muscle tissue in the heart, skeletal muscle tissue attached to the skeleton, smooth muscle, that's inside those tubes. That's the first thing we needed to know. The second thing we needed to know was contracting and relaxing our muscles. And this is where you kind of gotta, in the words of Sesame Street, my kids were watching Sesame Street this morning, you have to stop and think it through. So if we're thinking about our tube, and our tube goes from being wide to being much smaller, think about what you would have to do if you made a circle with your hands, and then you made that circle get smaller. That's the same thing that's happening with the bronchial tubes. You're contracting your finger muscles to go from a big circle to a smaller circle. So we had to consider what's happening with the muscle movement. Just like we dissected this question down, when you're taking your exams and you got those notorious application questions, dissect those application questions down too. Break them into small parts, think about them, figure out what answers you can get rid of right off the bat. Then we're just toying with 2 answers or 3 answers and keep working our way through eliminating them. So if we learn nothing else today, we have learned the way that a rescue inhaler works for asthma. That rescue inhaler tells the smooth muscle tissue that it needs to relax. If you do that, a person can breathe again. How do we feel about our group work activity for today? Thumbs up, thumbs down, any questions that we still have? Oh man, Ella Pina feels great. She's dancing. Yeah, Kaylin says she liked everything except the last question. I know. Tricky Dr. Ollis. I gotta make you think. All right. Well, let's dive in then to some of the regular lesson number 9 stuff. Let's see how much we can work through today. So as we start our lecture discussion of lesson number 9, we should do a little bit of lab review. Remember that, oh, and I'll mention too, by the way, help me out. We're looking at a picture of one of our types of muscle tissue. Which type of muscle tissue are we looking at pictures of? Can we tell which one this is? Yeah, that's right. We are looking at skeletal muscle tissue in these pictures. And here in lesson number 9, that's pretty much the only kind of muscle tissue we're going to focus on. So we're talking about the types of muscle tissue in general. That's what we hit here in the group work. That's all we're going to talk about. The other ones, we're really going to dive into skeletal muscle tissue. Audrey's totally right. We got all the myofibrils, right? So all of these protein tubes that fill up skeletal muscle tissue, those are called myofibrils. The myofibrils are made of things that we're going to talk about. We mentioned them in lab before. Things called sarcomeres that actually do muscle contraction. So these patterns that we see inside here, that's the parts that do muscle contraction. Remember when we talk about skeletal muscle tissue, that one of the things for you to remember that tells you it's skeletal muscle tissue is that beginning part, sarco. Remember, sarco always means muscle tissue. Sarco always means muscle tissue. Hey, just like chondro always means what again? What did chondro always mean? Yep, that was my cartilage word, absolutely. Does anyone remember the beginning part for bone tissue? What was that beginning part for bone? Yeah, exactly. Osteo, that's my bone word, right? So a little bit of terminology review. We're talking about sarco stuff today. The other beginning part that also means muscle, I'll add this to my list, is myo. If you see something that starts with myo, if you see something that starts with sarco, both of those are things that mean muscle tissue. So we're looking here at the sarcolemma, and here it is on this image as well, the sarcolemma. Remind me, what's the normal cell word for the sarcolemma? In every other kind of cell, we would call the sarcolemma, yeah, exactly. It's plasma membrane. It's plasma membrane, the sarcolemma. We also have, so it's abbreviated right here, SR. We type it out for us. Sarcoplasmic Reticulum. The Sarcoplasmic Reticulum. That's going to be, in a normal cell, what we call the endoplasmic reticulum. So we abbreviate that ER, right? The smooth ER and the rough ER. When we're in a muscle cell, since sarco is muscle, we call that the sarcoplasmic reticulum. We also have those things, myofibrils, that have those proteins inside of them. The one other thing to mention, we said this in lab about skeletal muscle tissue, is that skeletal muscle tissue has more than one nucleus. So we can see all those nuclei here in skeletal muscle tissue. The word to describe that is multinucleate. Multinucleate, the fact that skeletal muscle cells have lots of nuclei inside of them. Let's see if we can remember from lab. When I talk about this blue part here, the sarcoplasmic reticulum of a muscle cell, what was the function of the sarcoplasmic reticulum? Why is the SR so important for a muscle cell? Yeah, so the sarcoplasmic reticulum is where I store calcium, and it's where I'm going to release the calcium during contraction. When a muscle does contraction, we're going to spit out calcium from the sarcoplasmic reticulum. That's going to allow us to actually do contraction. I like that. Kaylin's tying in some of our stuff, way we've talked about things. The sarcoplasmic reticulum is kind of like my lacuna for calcium, my home, right? My special space for it. I like that. Okay, so we're storing all of our calcium here inside the sarcoplasmic reticulum. That sarcoplasmic reticulum, shown here in blue, is right next to these white tubes that you see right here. These white tubes are called T-tubules. T-tubules, quick review for us, are the parts of the plasma membrane, so the sarcolemma, that fold in. So it's places where the sarcolemma folds inside. This is going to be important, as we'll talk about today, because it's going to allow us to get a message for muscle contraction to go from one part of a muscle cell to the entire muscle cell. So allows us to tell the whole muscle to contract. That's the job of the T-tubules, to take a message that we receive in one place on the sarcolemma and send it all the way to the very middle of our muscle fiber, all the way to where those myofibrils are right next to that sarcoplasmic reticulum. There is at least one homework question that asks you to review the functions of these parts of a muscle fiber. That's our special name, by the way. I don't know if we use that in lecture or in lab. A muscle fiber is a muscle cell. So all of these are special parts of our muscle fibers. Make sure we know the functions of each of these parts and how they relate to the process of muscle contraction. Just another quick peek to show you all those myofibrils and the sarcolemma. My myofibrils are where we see the striations in a muscle cell. And if you look at these myofibrils, what I like about this little picture here, notice how we can see this little zigzag line right here, little zigzag, and here's another zigzag right here. This, in between these two zigzags, this is that thing we talked about in lab called a sarcomere, the contractile unit of a muscle cell. When we talk about a sarcomere that goes from one zigzag to another zigzag, what was the name of these zigzags here? Yeah, exactly. Yep, these are what we called the Z-discs. The Z-discs. So they zigzag back and forth, the Z-discs. In between them, in the very middle, is that red line. Let me draw a line to it. These red lines here in the middle of the sarcomere, yeah, exactly. Those are my M lines, M line. So when you're looking at a type of muscle tissue and you see the striations like we see here, so they got dark and light, etc., you're seeing the dark that's around that M line with all of the myosin, and then the light that's in the places where we just have actin, where we have that Z disc. And then we've got the dark where there's that myosin, and we'd have the light again where there's just actin. That's what striations come from. The fact that I've got these long patterns of these sarcomeres that alternate back and forth, back and forth. Yeah, exactly. Alpine is mentioning, and I think, let me see if I've got that picture. We'll come back to these. She's mentioning some of the terms that we used in lab when we were describing the parts of the sarcomere. So we'll label this one together. Let's hit the stuff we already did. These little zigzags here at the end, these are called the Z discs, like you already told me, those two Z discs. We've got that M line in the very middle of a sarcomere. Here, I'll label it on my bottom one as well, the M line. In lab, we just talked about this space right here. We labeled this space right here where there's only myosin. The place where I only have myosin, absolutely, like we're saying in the chat, it's called the H zone, where there's only myosin. But there's also places that we call this the thick filament myosin. There are parts of the thick filament that also overlap with actin, so we can see that here. And that area, so including the overlap, so from the beginning of myosin to the end of myosin, including that overlapping area that we've got in there, this whole space here is what we call the A band. The A band, absolutely. The places where we only have actin, so here on the out part, here where we only have actin, actin is the thin filament, so the thin letter I, the I band. Let's see if I can draw what I tried to explain in lab and didn't do a great job. So we've got our H zone. Oops, get my pencil here. We've got the H zone where I have the thick filament. We've got the I band where I have the thin filament. If we try to cross these letters with each other, we end up with an A in the middle. So imagine if we took these two little parts here of the H and we put them toward the middle where the I would be. That's how I remember. The A band has overlap between the H zone with myosin and the I band with actin. So the A band is what makes the dark part of a striation, and the I band is what makes that part look lighter when we're talking about striations. This sarcomere on the bottom part here has a pretty big I band, a pretty big space where there's only actin, none of the myosin that we see here. When we look at this sarcomere up here, my I band is much smaller, but I also am missing an area in the middle where there is just myosin. What part of my sarcomere is missing since I don't have any places here that just have myosin? Yeah, exactly. A couple of us have chimed in. If I have nowhere where there's not just myosin, I actually lose my H zone. So the space here in the middle where it's just myosin, that's gone when I do skeletal muscle contraction, because how contraction works is the actin moves toward the middle. The amount of overlap, the thing we call the zone of overlap, actually shrinks actually gets much larger when we're talking about contraction. The amount of space that we overlap, so the A band, gets much bigger when we're contracting, and it gets much smaller as we relax, as actin pulls away from myosin. So this particular sarcomere is an example of a contracted sarcomere. I know that because I have no H zone in the middle. The other way you can tell too is if you look at this picture, notice how there's actually some places where myosin and actin are interacting with each other. That only happens when a muscle is contracting. So contracted sarcomere, relaxed sarcomere. Let's back up briefly to talk about these two filaments. We already kind of talked about these in lab, but I especially want to take some time here with our thin filament. So the thick filament's really easy to remember. There's just one protein found in the thick filament. That one protein is myosin. So you should only ever tell me that myosin is the protein found in the thick filament. The thin filament, as we see in our picture here, is actually technically made of 3 different proteins. So most of the thin filament is these blueberry-looking proteins that we see here. These are the actin proteins. Actin is what directly touches or directly interacts with myosin, specifically with these little golf club head parts of myosin. These reach up and grab actin at these little myosin binding sites. So you can see those dots here. The myosin heads grab the myosin binding sites on actin and pull actin toward the middle. But we're not always contracting our muscles. It would be incredibly painful if we were. So we need to have some regulatory proteins, like it mentions here, regulatory proteins, to help us prevent contraction from always happening. So there's 2 of these, tropomyosin and troponin. Remember from lab that tropomyosin is the protein that covers the myosin binding sites on actin. So in other words, this special place where myosin likes to grab actin and pull it along so that we can do contraction, if tropomyosin is laying on top of those myosin binding sites, we're blocking that interaction. You can't contract if tropomyosin is covering up the myosin binding site. But here's the thing about tropomyosin. I like to think about tropomyosin like a wet spaghetti noodle. So think about this. My 4-year-old loves cold spaghetti noodles, of all things. So she likes those spaghetti noodles. If she holds it up, it's going to wiggle all over the place. It's going to go everywhere. If I'm trying to use a wet spaghetti noodle to cover my binding sites on actin, I'm going to need a little bit of help to make sure that it stays in the right place. That's the job of troponin. So troponin is these little yellow proteins that we see here next to those spaghetti noodles. Troponin acts like a pushpin. So it holds tropomyosin in place. As long as that pushpin is there, as long as that anchor is there, my spaghetti noodle is going to stay covering the myosin binding sites. And if it's covering the myosin binding sites, then myosin is not going to be able to attach. We will not be able to do the process of muscle contraction. When we talk about troponin, though, troponin has a weakness. Well, it's not really a weakness per se, but there's something about troponin. If something interacts with it, troponin, that pushpin, pops out. Yeah, exactly. A couple of us are mentioning in the chat, troponin, we would say, is calcium sensitive. Calcium sensitive. Meaning, if calcium attaches to it, troponin is going to change its shape. And when troponin changes its shape, this pushpin is going to pop out. And if I don't have the pushpin holding this wet spaghetti noodle in place, it's going to fall off and those myosin binding sites will be exposed. If those myosin binding sites are available, myosin and actin are going to attach to each other and we're going to do muscle contraction. If I cover them up, then we're not going to be able to do the process of muscle contraction. So big picture, when you're thinking about the function of calcium in muscle contraction, the goal of calcium is to change the shape of troponin, which will allow tropomyosin to move, which will allow myosin and actin to interact with each other. Yeah, so Elapina mentioned in the chat, so calcium is very important. Absolutely, yes. Not only do we need calcium for our bones, right? That's what we learned with rickets and osteoporosis. We also need calcium when we're talking about muscle contraction, because without calcium, these push pins are going to stay in place and we're not going to be able to do the process of muscle contraction. I didn't mention it before, but I did see Kira's hint. She says one of the ways that she likes to distinguish between tropomyosin and troponin is that tropomyosin is a long word and tropomyosin is a long protein. Yeah, so I like that. Tropomyosin, a long word and a long protein. It's the spaghetti noodle that covers the myosin binding sites. Troponin, that short word, that little small protein that acts like a push pin. I like that. Yeah. Yeah, so Audrey put a question in the chat that I get every single semester asking about troponin and heart attacks. So there are different types of troponin in different types of muscle tissue in the body. But when we talk about a heart attack, muscle tissue starts dying, cardiac muscle tissue starts dying, and part of that dying process is these troponin proteins are actually released. Yeah, I heard myself cutting out. Am I back now? I think. Maybe I'm not. Okay, back now. Okay. So when you have a heart attack, the cardiac muscle tissue starts to die. And as it's dying, we start to see these troponin proteins floating around. Troponins being my push pin that are holding that spaghetti noodle in place. If I can't block muscle contraction, my cardiac muscle is going to contract too much. So heart attack, an issue where the heart muscle isn't contracting correctly. To my understanding, I am not a pro at this, so talk to your A&P2 teacher about this. But to my understanding, what we're measuring in the blood is when troponin is falling off of cardiac muscle tissue, essentially. Let me see what else. Yeah, so Tian asked the question in the chat, if we've covered up that myosin binding site, does that mean our muscle is not contracting? Yes, that is correct. As long as I have tropomyosin on top of the myosin binding space, there's no spot for myosin to interact with actin. So here, let's bounce back to our sarcomere. If I have a little brown line, if I've got tropomyosin that's covering all of my places where myosin wants to attach to actin, it's not going to have anywhere to grab it. If I can't grab it, like we see happening at various places here in the sarcomere, if I can't physically attach to it, I'm not going to be able to do the process of muscle contraction. So I have to have that site available. If I don't, I cannot contract. That's absolutely correct. Yeah, so Audrey's kind of tying it into some of the terminology from a general biology class. Yeah, so the attachment site on actin in some ways is similar to what you would call an active site of an enzyme. So where stuff happens. We don't necessarily break anything down at the myosin binding site, but I do have to have that particular attachment site for myosin to be able to grab on, for myosin to be able to change its shape and cause the muscle to move. So in that regard, just like if we had an enzyme that we messed up its active site where it does a chemical reaction, it's the same deal with those myosin binding sites on actin. If I mess them up, if I cover them, I'm not going to be able to do contraction. So yeah, great connection to make there. So this brings us to a question. Can you help me out? What page in your notes do we have this question? I'm pretty sure I gave you in the outline this picture and this question. Okay, page 5. Perfect. Okay, so at the top of page 5, I am asking us to pause for a moment and to consider the big idea of our class. Remember way back on day 1, I asked you to think about the relationship between anatomy and physiology. Anatomy and physiology. When we talk about anatomy and physiology, remind me, these things are or are not related to each other. Anatomy and physiology, they are or are not related. Yeah, exactly. They are definitely related, right? They depend on each other. So when we're looking at the process of muscle contraction, when I am not contracting, so here we'll add a note for ourselves. When I am not contracting, the anatomy of my actin protein is that tropomyosin is covering the myosin binding sites. And troponin, my pushpin, is holding it in place. If this is my anatomy, if tropomyosin is blocking those sites, my physiology is that I can't contract. If this is my anatomy, where tropomyosin has slid out of the way, the spaghetti noodle has moved, because the pushpin, troponin, has also changed shape because calcium attached to it, big, long chain of events, right? Long story short, if I change the shape of tropomyosin, now those myosin binding sites on actin are available. If they're available, my physiology changes, and now my muscle can contract. Now I am contracting. So this question asks you to think big picture. When we're thinking about the appearance of our proteins, especially the actin protein and tropomyosin and troponin, when I think about their anatomy, that can help me predict their physiology. Are they contracting or are they not contracting? Just like if I told you they were contracting or not, you could predict their anatomy. Each semester, students get really hung up when they see this question. They're like, what are we talking about? All we're talking about, and all I want to make sure that you understand, is a contracted sarcomere. My thin filament looks different compared to a relaxing sarcomere. My thin filament looks different. Comes down to the shapes of my actin protein and its regulators. If I change the shape of the regulators, that's going to change what my actin protein looks like, and all of that's going to lead to a difference in physiology in the process of contraction. Muscle contraction happens on the smallest scale in a series of 4 steps. So this series of 4 steps, and this is something we want to underline, highlight, star. We need to make sure we understand these 4 steps. This is something called the cross-bridge cycle. The cross-bridge cycle. When we talk about the cross-bridge cycle, this is the way that we slide the actin filament past myosin. This is the way that those Z discs get pulled together. So let's go through and talk about each step and what happens in each of them. My first step is called cross-bridge formation. Cross-bridge formation. Now we should probably define, since it's in the name of this process, what a cross-bridge is. It sounds scary, but all a cross-bridge is the place where myosin attaches to actin. Where myosin and actin attach to each other, that's called a cross-bridge. So in step 1, a cross-bridge forms. Myosin attaches to actin. That's the beginning of the process of muscle contraction. Step 2 is the power stroke, or sometimes you'll hear it called the working stroke. The power stroke, the way I like to remember this one, is this is how your muscles give you power. So when you're doing a squat, we talked about legs yesterday, right? Movements of the legs. When we're doing a squat, the actual movement of lifting up with the weights, the movement that comes when you're doing a muscle contraction, that movement happens in this stage, the power stroke. Once I have moved actin past myosin, it's time for me to let go of actin and get ready to do this whole process again. Muscle contractions happen really fast, and there's a whole lot of attaching and detaching that are going on. So once I have pulled actin as far as I can, I need to let go of actin. And that's what's happening here in cross-bridge detachment. In cross-bridge detachment, myosin and actin let go of each other. This is a good thing for me, myosin and actin letting go, because if myosin and actin let go of one another, I can then go through my last step of the cross-bridge cycle called cocking the myosin head. Cocking the myosin head is changing the shape of that head from being bent over and not able to interact with actin to being into its activated, or what they call the upright position. When myosin is in this upright position, if those myosin binding sites are available on actin, it'll go straight through then and form another cross bridge. If we form another cross bridge, then we're going to pull on actin and slide it, then we're going to let go of actin, and then we're going to go back to that activated position. There's a great video in the guided lesson that shows you like a 3D modeling of how this process works. So this happens rapidly, these 4 steps. We just keep going and going and going. As long as those myosin binding sites are exposed, myosin will always attach to actin. We will always form a cross bridge. Okay, let's pause for a moment. When we talk about the cross bridge cycle, the first thing you need to know about each of these steps in the cross bridge cycle is what we just talked about. What's happening with myosin and actin? Are they interacting with each other? Are they changing their shape? Are they moving? That's the kind of stuff we need to know first. What's happening with myosin and actin? The next thing that we need to know is what's happening with myosin and its energy. So it should be no surprise to us that the process of muscle contraction takes energy, takes ATP. ATP is going to be broken up to allow us to harvest energy. And here's where I need to get a little chemistry heavy on you to help us understand what this stuff right here means. So let me get my pencil. When we talk about ATP, remember that ATP, here I'll type, stands for adenosine triphosphate. Adenosine triphosphate. Tri means 3, like a tricycle has 3 wheels, right? So let's draw a really basic, not chemistry accurate version of this. Adenosine triphosphate. Here's my adenosine, and then I've got a phosphate, and I've got a phosphate, and I've got a phosphate. Adenosine triphosphate. It's tri because there's 3 of them. 1, 2, 3. When we talk about ATP, or adenosine triphosphate, the way that we actually store energy in ATP is because of this chemical bond right here. This is my energy. I store it between ADP, where there's 2 phosphates, and an inorganic phosphate by itself. ATP, what I see right here, this is a molecule that has an adenosine with 3 phosphates attached to it. We've got lots of energy stored right here in this chemical bond. When myosin breaks this chemical bond, so let's chop it up. When myosin breaks that chemical bond, we end up with adenosine diphosphate. Adenosine diphosphate. Adenosine diphosphate doesn't have this high energy bond out here. So there's not energy in ADP. Put that abbreviation down here. ADP. Here's where all that energy was in ATP. If I chop up that bond, there's no more energy being stored. However, that energy is what I use to take myosin from being bent over to being in that upright position. So notice when I detach from myosin, or from actin, excuse me, that molecule of ATP is attached to me, I'm holding on to it. If I chop up this ATP molecule, if I steal that energy, now I can stand up and I'm left with ADP, so this molecule that has 2 phosphates, and then I've got a random phosphate group that's just chilling out on my back as well. So here's a note for us to write for ourselves. This step right here, cocking of the myosin head. This is the step where I do ATP hydrolysis. ATP hydrolysis. Let's put that in easy words. ATP hydrolysis. Breaking up ATP using water. Hydrolysis means water breaking. So the way I do it is I would, remember how, here's the tie-in for us. Remember how we talked about dehydration synthesis? Dehydration synthesis. This was when I built a chemical bond by taking out water. That's how I built those peptide bonds. Yeah, like Elapina mentioned. I can actually do the opposite of dehydration synthesis. It's called hydrolysis. When I break up a chemical bond by giving back a molecule of water, that's going to release some energy. So in this cocking of the myosin head step, we chop up ATP to get its energy to help myosin get back into the upright position. So when you're looking at the homework assignment and it asks you which stage of the cross-bridge cycle we break up ATP or we do hydrolysis, when do we actually get that energy, release that energy that's stored in ATP? That's in this step right here. We chop up ATP in the cocking of the myosin head step. Because when I cock the myosin head, getting it standing upright again, that takes energy. When I'm standing in this upright position, I'm attached to ADP, I'm attached to inorganic phosphate, because I broke them apart to get my energy to stand up. When I attach, I can then go up and attach to actin if its myosin binding site is available. With the myosin binding site available, I'm attached to it. Myosin and actin interact with each other. When I attach to actin, if I'm myosin here, I say, whoa, this is awesome. I love actin. I don't need any more of this junk attached to me. There's no more energy left for me in ADP. Inorganic phosphate is absolutely a leftover. So I spit out, or I detach from ADP and inorganic phosphate. When I let go of those things, that changes my shape again. So in this stage right here, in cross-bridge formation, when I first attach to actin, I'm still holding on to the trash that's left over from breaking up ATP. But now I've got a new girlfriend, right? Actin is my new main squeeze. Actin's jealous, says get rid of those things. We spit out ADP and phosphate. When that happens, we do the power stroke stage. Because I no longer have these molecules that are keeping me positioned upright, I'm going to bend over. I'm going to move actin and actually make energy. So the step where myosin releases ADP and inorganic phosphate, when I let go of those things, that allows me to pull actin. That allows me to make movement. But once I've spit these out, and once I've bent over like this, I'm kind of stuck. I'm attached to actin. I've got nothing else to change my shape again. I don't want to stay attached to actin. I'd like to be released. What allows me to let go of actin so that I can get back into the upright position and pull again is when a molecule of ATP attaches to me. So in the cross-bridge detachment step, remember that's when we said that actin and myosin let go of each other? What actually makes actin and myosin let go of each other is when a molecule of ATP comes and attaches to myosin. Myosin was like, actin, I love you, but I found someone better. I found ATP. So ATP attaches to myosin and causes myosin to let go of actin. But if myosin is attached to ATP, myosin realizes, ooh, there's some energy in here, and we break down ATP to go back into the upright position. We talked about at the beginning how we need to know what's happening with myosin and actin in each of these steps. But the other thing we need to make sure we know for each of these steps is what's going on with ATP or what's going on with the trash that's left over from ATP, when I do hydrolysis. I attach ATP to myosin to get it to let go of actin. I chop up ATP to help myosin go back into the upright position. If myosin is in the upright position, it will automatically attach to actin and it'll pull actin along. But we run into some trouble if I never have ATP to keep me going through this cycle. See, there's this thing you're studying in this lesson called Rigor Mortis. Rigor Mortis. For my friends who've had a chance to work on this activity, what is rigor mortis? Yeah, Elapina's mentioning it has to do with dead bodies. Absolutely. We get to talk about all kinds of fun things this unit. Dead bodies. Yep. Okay, so absolutely rigor mortis. So that rigor word means stiffness and mortis means death. So rigor mortis is when the body gets stiff after death. Body gets stiff after death. I like it. Kaylin called it creepus maximus. I like that. Okay, so rigor mortis, if you ever watched, for a while, I was really into the TV show Bones. Any Bones fans out there? This was a while back. It's been off air for a little while. Yeah, Lapina's laughing at me. Fanshin liked it. Yep. Hey, here's what I'll tell you. At the end of anatomy class, once you've taken your lab final exam, you should go back and watch a few episodes because they will talk about bone markings that you learned. So you'll feel way smarter watching bones after you finish this class because we're learning all about those bones. So we can have a bones watch party, right? That'll be the other thing we do. After you all come over to have our salty bananas and do homework on my porch, then we can all watch bones, right? Okay, so rigor mortis, when your body gets stiff after you die. When you die, do you continue to make ATP or do you stop making ATP when you die? Yeah, so when you die, Caitlin says, I hope we don't keep making it. That would be really creepy. Yeah. When you die, you stop making ATP. See, the way that your body makes ATP is we use oxygen that we breathe in, and we make a lot of ATP using that oxygen. When you die, you stop breathing, you stop making ATP. When we talk about the body getting stiff after you die, the body getting stiff after you die happens because you're not making ATP anymore. So let me make that note for ourselves here. This happens because we have no ATP. Rigor mortis happens because we have no ATP. I need you to help me remember, what is the job of ATP? What does ATP actually do in this cross-bridge cycle? Okay, so it is my energy. That is correct. I do use it as an energy source. Before I use it for energy, what do I use it to help me do? Yeah, so here's where I use it for energy. When I cock the myosin head. Yeah, there we go. Kira mentioned it. The first step that you actually use ATP, the first thing you actually use ATP to do is this. Cross-bridge detachment. Remember, I told you that when we finish the power stroke, myosin is attached to actin, but it's stuck. What makes myosin not attached to actin is when ATP comes along and attaches to myosin. If ATP attaches to myosin, myosin lets go of actin. I can't do cross-bridge detachment. I can't get myosin and actin to let go of each other unless there's a molecule of ATP available. Okay, so I'm going to put this on you all. Can we figure out, why do my muscles get stiff when I die? What's actually happening? Yeah, there we go. Kira got it for us. Rigor mortis, the problem is we did the power stroke, we are attached to each other, but we can't let go. If myosin and actin let go of each other, they can slide past each other. We can still do movement. But if I don't have ATP, I can't detach myosin and actin from one another. They're stuck together. So think about if you stick one of your hands pointing upright, like it's myosin, and you make your other hand on top of it like a T, we're stuck. You can try to wiggle your hands back and forth, but as long as your two hands are attached to each other, there's not a lot of movements you can do. That's what happens in rigor mortis. Myosin gets stuck attached to actin. I can't release them from one another, so my muscles get locked up, basically. They're literally contracted in whatever position they were last time I had ATP. Rigor mortis is an issue with this step right here, with the ability of your muscle cells, or the actin and the myosin in your muscle cells, to let go of each other. If I don't have ATP, they're glued together and I get stiff, or I experience rigor mortis. I need to pause for dramatic effect, but also because I need to give you a moment to process. Please help me out in the chat. What are we thinking? If you want to try to re-summarize what we just said, send me an emoji. I want to give us some time here to think on this. Yeah, Audrey said she's planning to rewatch this. That's a great idea. Kaylin has all the emotions. ending with her mind blown. Hey, one of your classmates, I will toss this out here. One of your classmates offered me a really great study idea. She said, as we get closer to the unit exams, listen to these class sessions at 2 times the speed, because you'll still review all of the content, but it'll go a little bit faster. The time goes faster. So she said every time when we get closer to exams, she rewatches the class sessions at 2 times speed. So when you're in YouTube, there is, I think it's in the upper right-hand corner, you can change how fast it plays the video. Oh, it's bottom right. Thank you, Kira. Kira knows it better than me. I don't do a lot of YouTube. So bottom right corner of a YouTube video, you can click on the little thing that looks like a gear, and it'll give you the option to play it back faster. Or you can play it back slower too. I don't know why you'd want to listen to me talk any slower, but you know, maybe you do. So you can change that speed on the videos and listen to them a little bit faster. Get the stuff again. Probably don't want to listen to this faster just yet, right? Oh, that's funny. Audrey mentioned that she had an art professor that talked a little bit slow, so she had to speed him up. That's great. Yeah, Kira's speeding through her micro lectures. That's awesome. Okay, we will, when we do lecture again next week, I am definitely going to give us a group work activity that asks us to apply these steps of the cross bridge cycle. But here is my study pro tip for you as you're working on learning these things. Remember how I told you at the beginning of class to stand in front of a mirror and talk to yourself, or to find a classmate and talk to a classmate about this stuff? This would be one of those things that it's going to be really important that you be able to explain. We hate those explain learning objectives, but I'm just going to be honest with you. We got to know what's happening in these steps. Because you know how Dr. Allis is. Dr. Allis will say, well, what happens if your body doesn't make any ATP? Where are we going to get stuck? Or what happens if I can't chop up my ATP? Where am I going to get stuck? So you need to be able to explain what's happening in cross-bridge formation, what's happening in the power stroke, what's happening in cross-bridge detachment, and what's happening in this cocking of the myosin head step. So here's the list of things we want to know for each step. For each step, here we go. We want to know what's happening with myosin. We want to know what's happening with actin. We want to know what's happening with ATP. Or if ATP is not there, we want to know what's happening with ADP and inorganic phosphate. And Kaylin mentioned we'll add a little note for ourselves too. We would also like to know what's happening this week. What's happening in class. We would like to know that too. So when you're outlining your steps, when you're talking it out to the mirror or to each other, or just writing it down for yourself, if you are a read-write learner, you can totally just outline it yourself. That's totally fine. Make sure you can tell me if myosin and actin are attached to each other or not. Make sure you can tell me if they're sliding past each other, like we see in this step. Make sure you can tell me when ATP comes into the picture. So remember, we use ATP in this cross-bridge detachment step. And then we start to see ADP and inorganic phosphate in the cocking of the myosin head once we've chopped up ATP. So Fanshin asked if I had given you the question, right, where if I told you we can't chop up our ATP, what's going to happen? What's going to happen is we're not going to be able to do this step right here. We're not going to be able to cock the myosin head, so we can't get it back into the upright position. And if we can't get it back into the upright position, it's not going to be able to attach to actin again. If it's always stuck in this bent position it's not going to be able to reach up and grab actin. So ATP initially is needed to make me let go of actin, but once I chop it up, then I can use that energy it had to form a cross bridge and actually attach to actin again. I know this is a lot. Are there any questions that we can put to words right now as we're still processing? Totally fine if we can't put anything to words at the moment. We've been way too serious in class today. I need some emojis. This has been hard stuff. So I need some emojis. Shoot me some emojis. Yeah, Audrey asked when we're gonna get the anatomy cartridge that we can just plug in, right, and just know all this stuff. I'm sure there would be a market for it. I actually, my husband had to run to Target recently, and he asked, "Is there anything else that we need?" And I had texted him back, "We need sleep. You need to buy us some sleep." And he was like, "I'm sure if it could be sold, Target would sell." So I'm sure if anatomy could all be put into a cartridge that you could plug into your mind, you would probably be able to buy it at Target or Amazon. It'd probably be a little bit cheaper at Amazon. Just being real. Yeah, Fanchon would buy sleep too. I guess that's 5-hour energy, right? It's liquid sleep, or so they say. I drink a lot of coffee, but I don't do energy drinks. That's where I draw the line. True, yeah, vitamin B12. I've heard some good things about that compared to excessive caffeine. This is as far as I had asked you all to go to get ready for class today. Light me up in the chat. How many of you were able to work past this? Did any of us get past this? Okay, a couple of people have. I'm going to keep going. Even if you haven't had a chance to work on the new stuff, that's totally fine. We'll cover some of this stuff. That'll give you a head start as you're working on the homework assignments. I believe, I can't remember for sure, but I believe this is where the part 1 homework assignment ends. So you should be able to do lecture part 1, that homework assignment. I think we've covered now all of the material on that. So that's good news for you to help you stay on track. You should be able to get that one done. Let's take a step back. So when we talk about the process of muscle contraction, what we just talked about is what you see down represented here at the bottom of my picture. We just talked about the cross bridge cycle. The process where myosin attaches to actin, and we actually do the process of muscle contraction. The ability of myosin and actin to form the cross bridge and to interact with each other actually has to start all the way back, much bigger. So we take a step backwards, we're here, and we take even one more step backwards out here. The process of muscle contraction actually starts at the sarcolemma, where a neuron sends a message to a muscle fiber telling it to contract. So the ability of you to do the cross bridge cycle, for you to actually contract muscles, you have to get a message from a muscle or from a neuron to tell you to contract. So big picture, when we talk about muscle contraction, we're talking about something called excitation-contraction coupling. That's biggest picture. How does muscle contraction work? Biggest picture how it works is what we call excitation-contraction coupling. Now, this sounds really scary and really crazy, kind of like the central dogma. Let's break it down so it's not quite so scary. We just talked about contraction, right? So this is the cross bridge cycle right here. The way that a muscle goes from being long to being short. This excitation part tells us that how we get contraction to happen is by changing the membrane charge. Changing the membrane charge. Okay, remember way back in unit number 1 when you hated me for talking about gated channels. Guess what's back? Those gated channels. They're back to haunt us. Because I'm talking about changing my membrane charge. If I change the charge of my membrane, I can make stuff happen. Well, the way I change my membrane charge is by using different kinds of gated channels. So let's talk about these things. Up here at the beginning, I've got my motor neuron. My motor neuron talks to my muscle fiber using special chemicals called neurotransmitters. Remember those from Unit 1, neurotransmitters? When we're here in Unit 3 and we're talking about the way that neurons talk to muscles, there's one particular neurotransmitter that neurons use to talk to muscles, and that's called acetylcholine. Acetylcholine. So you see it abbreviated there, ACH, acetylcholine. This would be an underline highlight star term. I'm not going to make you spell it, but I want you to know this is the one message that neurons send to muscle fibers. This one message, acetylcholine, my neuron spits it out. That message goes across that thing called the neuromuscular junction, like Elapina mentioned. We highlighted in lab, remember, the neuromuscular junction, where a neuron talks to a muscle cell. So the neuron spits out acetylcholine. Acetylcholine goes down and attaches to some ion channels that I see here in the membrane, in the sarcolemma of my muscle cell. Okay, not a trick question. These channels that I see right here, they are gated. If I'm a gated channel, I am or am not always open. If I'm a gated channel, I am or am not always open. Yeah, exactly. I am not always open. That's what gated means, not always open. When I open because of acetylcholine. Acetylcholine opens me. So acetylcholine opening me means I'm what kind of gated channel? What kind of gated channel do I have if a neurotransmitter opens me? Yep, check that out. Chemically gated, and I'll tell you, these are sodium channels. Chemically gated sodium channels. A neuron wants to tell a muscle fiber to contract. So the neuron spits out acetylcholine. Acetylcholine goes down and attaches to a chemically gated sodium channel. Chemically gated sodium channels with the right chemical key, which is acetylcholine, open up and sodium can move. Hey, guess what? We get to talk about the salty banana again. It's back. I missed it in unit number 2. Not really, right? We didn't actually miss it. But it is back. We're going to use it all kinds of crazy here in unit number 3. Let's draw a banana for old time's sake, because we haven't done this in a while. Here comes my salty banana cell. Remember, for our friends who are not as familiar with this, the salty banana helps us to remember what I have a lot of inside and outside. So we're talking about sodium. We're opening up channels for sodium. Where do we have a lot of sodium when we're talking about our salty banana, that banana that we sprinkled salt on? Yeah, lots of sodium on the outside. There's my sodium. Salt, by the way, is sodium chloride. So I've got a lot of sodium and chloride on the outside. And just like a banana, what do we have a lot of on the inside? What is there a lot of inside a banana and inside our cells? Exactly, potassium. Okay. We're not going to talk a whole lot about chloride in lesson number 9, but we are going to talk about it in lesson number 10. So don't forget about chloride. We're going to use that with our neurons. When we're talking about muscles, we are primarily going to be concerned about sodium and potassium. These are chemically gated sodium channels. When I open up a channel protein, things always move from high concentration to low concentration. That's the way channel proteins work. We always go from high to low. So if I open up a channel for sodium, is sodium going to go out or going to go in when I open up a channel for it? Yeah, exactly. Sodium's going to go in. Hey, remind me, sodium, is that a cation or an anion? There's a unit number 1 word. Cation or anion? Yes, exactly. Sodium is a cation. I should have put a little plus on it. We'll add a little plus. This matters that sodium is a cation because when I let sodium inside my muscle fiber, the charge on my sarcolemma starts to get positive. When my charge starts to change, what kind of gated channels start to open if the charge is being all funky now? Yeah, exactly. My poor baby. I apologize. You can probably hear him. When the charge changes on the plasma membrane, we start to open voltage-gated channels. Voltage-gated channels. Now, when the charge first starts to go up, we're going to open other sodium channels that would help the charge continue to go up. When we get all the way up, kind of like we talked about with neurons, when we get all the way up, we're actually going to open up potassium channels to help us come back down. And we'll talk more about those in a bit. Right now, what we care about is our voltage-gated sodium channels. Voltage-gated sodium channels are what you find all across the sarcolemma of a muscle fiber. So say my neuromuscular junction is over here. Let's draw a little line. We'll pretend that our membranes are attached here. Say that this is the place where a neuron talks to my muscle fiber, to my muscle cell. And this is all one big cell here. If I talk to my muscle cell right here at the neuromuscular junction, all the way between right here and all along this membrane and all along here, we're going to have voltage-gated sodium channels. The job of the voltage-gated sodium channels is to help my membrane charge change everywhere. Because see, if I change the membrane charge right here, that makes this area start to get a little positive, which will make me open up those channels here, which will make this part get positive, which brings in enough sodium to make this part a little positive. We're generating something that's called an action potential. Action potential. Think of an action potential like a message that moves. When we talk about a cell like a muscle cell or like a neuron, messages are always travel in the form of charge or of voltage. So when I change my charge at the neuromuscular junction, I use a voltage-gated channel to change this part of the membrane, which will help me change this part, and we go all the way down the membrane, all the way across the membrane. All across the sarcolemma, we've got voltage-gated sodium channels that start to change the membrane charge and move that charge various places. But then remember these guys here, those things called the T-tubules? When we talk about T-tubules, these are places where the plasma membrane actually goes into my muscle cell. Yeah, so we folded in my membrane. Lining my T-tubules, I still have those voltage-gated sodium channels. So I can keep changing my membrane charge. I can keep doing this action potential thing. But the other thing that we have inside these T-tubules is what you see right here. It says a voltage-sensitive tubule protein. Voltage-sensitive tubule protein. Voltage-sensitive means I change my shape when the charge changes. These particular proteins live right next door to the sarcoplasmic reticulum. The sarcoplasmic reticulum, remember, was the place where we stored calcium inside of our muscle cells. On my sarcoplasmic reticulum, I have some channels, some release channels. These channels will release calcium when they are pushed open. So these channels are not always open. If I'm not always open, I would be called gated, right? So this calcium release channel is a gated channel, and I have to be pushed. So like Kira mentioned, these channels are what we call mechanically gated channels. To get calcium out of the sarcoplasmic reticulum, I push open mechanically gated calcium channels. Mechanically gated calcium channels. They have to be pushed. That's what that word meant, right? We push on them. To get these channels pushed open, I use a protein that changes its shape when the charge is different. So what pushes on these calcium channels is this protein that lives in the membrane of the T-tubule. It changes its shape and pushes open the calcium release channel. When I push that channel open, out goes my calcium. And remember, calcium is what I used inside my sarcomere to get rid of that push pin. So before we can do that stuff that we talked about earlier, the 4 steps of the cross-bridge cycle, we got to do all of this stuff first. I've got to get calcium released so my push pin will pop out. The way I get calcium released is by pushing on my mechanically gated channels. The way I get my mechanically gated channels to be pushed open is to use something that's listening for the charge on the membrane. And the way I make the charge wrong is because of my voltage-gated channels that moved that charge, that started way back at day 1 with chemically gated channels. The chemical that starts this process, acetylcholine. The neuron spits out acetylcholine, opens up these little channels, a little bit of sodium comes in. That little bit of sodium opens up the voltage-gated channels. The voltage-gated channels move the charge all across the membrane until we get to those T-tubules. When that charge gets into the T-tubule, it changes the shape of the voltage-sensitive protein that pushes on my mechanically gated calcium channels. I suspect our minds are blown. Am I accurately reading the crowd? the virtual crowd. Shanti says, exactly. Kira's mind is okay. Hey, that's good. Yeah. Okay. Well, I don't want to cover anything else today because I want us to really think, kind of pause and rest on this as we wrap up our time together. Elapina asked a good question. Let me see if we can figure it out as a class. When I push open my mechanically gated calcium channels, what's going to happen next? What happens when I spit out that calcium from the sarcoplasmic reticulum? What's it going to do? Or where's it going to go? Yeah, there we go. So Kira told us specifically it's going to go down to attach to troponin. Once it attaches to troponin and pops it out and makes tropomyosin move, then like Fanchin mentioned, we would start the cross-bridge cycle. Remember when we looked way at the beginning of class today at the cross-section of a skeletal muscles fiber, how the entire muscle fiber was filled with our tubes, our myofibrils. Those myofibrils are made out of sarcomeres, of myosin and actin. So I spit out the calcium and it just goes into the space inside the cell. Friendly reminder for us, I think we use this word in lab, the inside of a cell, the soup and the organelles, called the sarcoplasm. The sarcoplasm, kind of like the cytoplasm that we talked about in a normal cell. I spit out my calcium into the sarcoplasm and it's going to float around inside the cell and go down and find some troponin. Yeah, so Alapina asked whether or not it goes into the T tubules. The calcium does not go into the T tubules. The calcium is going to go into the cell. So there's this little space you can kind of see here. The T tubules have a protein that pushes on a channel that allows calcium to go into the sarcoplasm of the cell. So my mechanically gated calcium channels, they are found on the sarcoplasmic reticulum. My voltage sensitive pushing proteins, if you will, that's found on the T tubule. But when they press on the calcium channels, the calcium just goes into the sarcoplasm. It's not going to leave into that T tubule. It's just going to go into the sarcoplasm. So the question says, the message to release calcium goes into the T tubule. Yes, that's correct. So my message to release the calcium is what starts over here with my membrane charge starting to change. And it travels all across the membrane down into the T tubule. That message causes the voltage sensitive protein to change shape, which causes it to push on my mechanically gated channel, which causes me to spit out my calcium into the cell. Yeah, so the voltage sensitive protein is in the T tubule, but the mechanically gated channel is in the sarcoplasmic reticulum. Exactly. So let's recap. We have talked about the 3 types of muscle tissue, talked about what they look like and what their general jobs are. We have talked about the names of the parts of a muscle cell. We've talked about the arrangement of the proteins inside those sarcomeres and how they slide past each other in the cross-bridge cycle. And then we ended with this idea of taking a message, excitation, taking a message, and turning it into muscle contraction. We talked about a lot of different things today. Again, most of this content is on lesson number 9, part 1 assignment. So if you have a chance in the next couple of days, try to start working on that. Obviously, remember that today we've got a couple of visible body assignments due for lab, so don't forget about those. You've got the lab assignments in Blackboard due on Sunday. This stuff isn't due until next Friday or next Sunday, I can't remember. But try to get on it. Try to get working on lesson number 9 if you have a chance. Yeah, the 4th, Vanshin's saying. I can't remember if that is Sunday or Friday. It is due on the 4th. That sounds like Sunday, probably. So we will wrap up lesson number 9 next week, again on Thursday. We will do next week's lab on Wednesday. Office hours, so student hours, on Tuesday and Friday. Please feel free to stop by if you want to chat about any of this stuff, or if you want to look over your unit number 2 exam. I'd be happy to chat through that with you. Otherwise, have a great weekend. I look forward to seeing you all back in class next week.